Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Fall Monday Seminar Series. Before introducing today's speaker, there's just a bit of housekeeping to tend to. So if you have any questions today for our speaker, please ask them using the Q&A icon located on the webinar toolbar. You can add questions at any time during the presentation. There's no need to wait until the end. And please ask your questions with the Q&A icon, not the chat box. And now I have the pleasure of introducing today's distinguished speaker, Dr. Andrea Baccarelli. Dr. Baccarelli is the Leon Hess Professor and Chair of the Department of Environmental Health Sciences at the Columbia University Mailman School of Public Health. He also serves as the Director of the National Institute of Environmental Health Sciences P30 Center for Environmental Health and Justice in Northern Manhattan. He also serves as the president of the International Society for Environmental Epidemiology. And Dr. Baccarelli is also the director of the Career Mode Program, which is an innovative nationwide NIH-funded program that supports early-stage biomedical researchers with mentoring, networking opportunities, and training in omics, data science, grant writing, and lab leadership and management. And along with his many other awards and honors, Dr. Baccarelli is also a member of the National Academy of Medicine. And finally, his research investigates molecular mechanisms linking environmental exposures to human disease and aging, which he will highlight today in his presentation titled Omics, Big Data, and Environmental Determinants of Aging. And with that, let me turn the screen over to Dr. Baccarelli.
Thank you so much for the invitation. It's amazing to be here and see many of you, many I already know. Today, I would like to give an overview of some of the items we do in our research and in my lab. But let me start with a general introduction. I strongly believe that these next 10 years are going to be the years of the exposome. We have been working a lot on the genome, and my research has been a lot on epigenetics, as you will see. But more and more, we have the realization that environmental exposures are increasingly important in our science, perhaps because of the limited capacity we have to predict and treat disease using genetics. Now we are realizing that the acquired risk factors are particularly important. And I would like to take a moment to define what exposome means, at least in my language. It is the equivalent of the human genome, and it's the attempt to measure and assess the totality of exposure that an individual experiences over their lifetime, and how these exposures affect health. And you will understand immediately how this is an ideal goal. While we know that a human cell has 20,000 to 25,000 genes, depending on how we count them, we don't have a head count of all the exposures we are exposed to. And more important, even if we knew about all the exposures that come at us, these exposures change over time. Studying genetics is easy or easier because genes rarely change. The DNA, the sequence of a gene rarely change. But the acquired risk factors around us, whether these are social, chemical, physical, nutritional, and so on, they change over time, and it's particularly challenging to measure them. So, however, they are particularly important. There are different estimates about how much of human diseases are caused or attributable to environmental causes. And we might think that this amount varies between 70 and 90%. And I want to remind ourselves that that doesn't mean that the genes only have influence on 10 to 30% of disease, because genes and environment are not mutually exclusive. There are many human diseases that are caused at the same time by genetic predisposition and environmental factors. However, it's really interesting that when we look at age-related conditions, we notice that the heritability of these conditions is relatively low. I mean, for instance, especially in conditions that happen later in life. For instance, if you look at death from a cardiovascular disease, from chronic ischemic disease, the heritability varies between 57% or 38% in men and women. Alzheimer's is a little higher, but Parkinson's, for instance, is very low. And hip fracture in people is still interesting if you look at this age strata. Hip fractures in people that are younger are highly heritable or have a certain amount of heritability. But if you look at people age more than 79, the heritability is very, very low. So it appears that at least on some of these conditions and diseases, the heritability becomes less important, the genetic component becomes less important as we age. And you can understand how for many diseases and many conditions, it's not unusual to see heritability estimates that range between 60 and 90%. So all of these are relatively low. And again, even if genes have a role, even for instance, in the 79% of cases for which there is evidence of heritability, there might still be an environmental exposure. And in fact, it's very likely that environmental exposures play a major role. So what I do in my lab is to attempt to capture the impact of environmental exposures of any type on our cells. And particularly over the past few years, my research has evolved in using molecular biology and data science to create biomarkers, particularly in blood, that you can see vials here all over the screen, that can fingerprint exposures. And by fingerprint is an analogy, is the idea that we can create and use a biomarker that is sensitive and specific, like our fingerprints belong only to our cells. So this might seem like a tall order, but in reality, we are lucky. We are lucky because environmental exposures already leave many types of fingerprints on human cells. Whenever there is an environmental exposure we are exposed to, our cells react mostly to cope with the exposures, to make us adaptable to the exposure and help us to adapt and survive. And that means that there are a lot of changes everywhere in our cells that are really fingerprints of that exposure. And we believe that some of these changes can be specific enough to create a biomarker that helps assess exposures and help detect the impact of exposures on our cells, our biology, and our bodies. So for instance, we published this review article a few years ago in Cell, where we used a very popular classification of hallmark of aging that is reproduced here. This is a figure that is very popular, many of you might be familiar with, that we adapted for this paper, where we identified 8 different molecular substrates that can be modified by environmental exposures and be important for aging. And we have mitochondrial dysfunction, epigenetics, I'll speak a lot about this today, genomic mutations and alterations, ox stress and inflammation, and so on. And this really, we are really in luck, because this is how we can use these changes that happen in response to environmental exposures to create new biomarkers. However, there are challenges, and I identified two challenges today that we have been working on and we have been trying to tackle in my lab. And I will go over, I have two sections of my presentation that will review these two challenges and possible solutions. And challenge number one is time. So I just mentioned that the exposome is about the totality of exposure individuals experience over their lifetime. The issue is that when we study people in our, particularly in my population-based studies, we don't have that type of data. We start to assess volunteers at a certain age or a certain time in their life, and we typically have only a few years or a few decades worth of exposure. So how do we actually go back and measure many more years of exposure? How we can ideally reconstruct the totality of exposure over someone's lifetime? So I want to propose a solution today, which is epigenetics. Epigenetics is about changes in the DNA, particularly in DNA function and gene expression, that are not related or due to alterations of the DNA sequence. A typical type of change is DNA methylation. Is a chemical modification of the DNA. It is added on top of the DNA sequence, but doesn't change in itself the sequence. So the reason why epigenetics is interesting to reconstruct the time and cumulative exposure over time, or is particularly because our cells may record memories in the epigenome. So our cells tend not only to change gene expression when there is an external exposure, but they also leave a mark that remind the cells about that exposure and about how to go about in responding to that exposure. So in response to certain exposure, there are changes in DNA methylation that continue to exist even after the exposure is removed. This is all about gene expression. We have 20,000 to 25,000 genes in our genome, and only 10% of those are switched on at any time in any of our cells. And this number remains in average the same, that there are many changes across tissues, across cell types, and many changes over time. And we are really lucky that our cells know how to switch them on and off. I'm always in awe to think how many things happen in our body that we don't have to worry about, that we don't control, and we don't even know about. And control of gene expression is one of them. There is a highly sophisticated framework that keeps genes switching on and off over time to help us adapt to changes around us and inside us. So, but I would like to walk you through an example to review together how we may use the epigenome to assess time. And it's an example, it's an analogy, which is a musical example. So what you see here on the screen is a music score. And in my example, this is the DNA. It is the DNA because, of course, it's a code that a musician can read that signifies music. So if you get this code, in the same way the DNA is the code, you can get a phenotype, which may be a symphony that you might want to enjoy at the theater any time of your day or your year. And The DNA is exactly the same situation. It's a code that codes for all of our proteins that make the symphony of our bodies appear and continue to live. And you can understand how in music, from the same book, from the same music score, you can really get many different phenotypes. Yeah, depending on the orchestra, depending on who's the soloist and the director. There is so much that can change, and so much that you can appreciate changing between performance and performance. And of course, I mean, this is something that we know to happen in genetics. With the same DNA, think about identical twins, we can have different phenotypes. Even identical twins that look exactly the same, they can have differences in the way they do over time, particularly as they age. Their health history can start to diverge, and one can get perhaps Alzheimer's and the other may not. One can age graciously and the other may not. And all of this is about genetics. I haven't told you yet anything in this example about epigenetics. In fact, and this really fascinates me, anytime I go to a concert, I make sure to go close to the stage and look over the actual music score that the director and the performers are using. And you typically see something more than just the score. You typically see the score still printed out, but you see also markings that are added onto the score that were not there. They were not part of the original score, but they are added by the director or the performers while preparing for the concert, just to remind them how to read the code, how to read the music. And the same, the cells, these are, the epigenetics are instructions, are annotations added onto the score, onto the DNA, that remind the cells how and when to switch a gene on and off. And really interesting, both in music and epigenetics, we might think about different types of annotations. We might have annotations in ink, the ones that are applied, they remain forever. And we have been aware, for instance, for many years that when the embryo develops, lineage commitment that lead to brain cells becoming brain cells and muscle cells becoming muscle cells are driven or at least associated with epigenetics. There are changes in DNA methylation that mark which genes are going to be active in brain cells and not in muscle cells, and vice versa. And these are in ink. Once they're applied in embryo life, they're very difficult to erase. There are, however, others that are more transient, like in pencil, and they can be classified, perhaps, in my imagination at least, in hard and soft pencil. And you can think about different type of situations. One that is easy to describe on marking in soft pencil are all the changes in gene expression that happen in the immune system in response to a microbe. A microbe comes in and the immune system needs to react promptly. And there are changes in DNA methylation and other mechanisms that happen in a matter of minutes. And typically the ones in soft pencil are applied fast and they go away fast. And vice versa, the one in ink are applied a little slowly and they can go away often slowly. But let me get out of the analogy now and discuss epigenetics itself, in particularly the mechanism that I study the most in my lab, which is DNA methylation. As I mentioned, DNA methylation is a program that controls genes. Typically, we tend to believe, though there are many exceptions, that when a gene is methylated, that gene is also switched off. It doesn't make RNA, it doesn't make protein. but there are also other situations, and it happens, it's an addition of a methyl group, CH3, on the cytosine, typically a cytosine followed by a guanosine, CG, or CPG better. And the advantage of DNA methylation in our study is that it can be easily measured in DNA, which is very durable, and it's very easy to get DNA even from a drop of blood. So we have been doing this for a while, and we have used a lot, and we still use this microarray that you see here, which is the Illumina EPIC array. Each of the slots here welcomes a DNA sample. So if you have the DNA sample from an individual, you will put it in one of the slots. And the array measures 850,000 methylation sites. So let's say there are 20,000 genes in our genome. You have about 40 methylation sites measured on each of the genes, which is far from being all of them that we have. We actually have about 20 million methylation sites on our genome, but it's a good compromise between throughput and feasibility. And particularly, these arrays are rather reliable. So we still use microarrays as opposed to sequencing, because the microarrays are easy to measure, are easy to use, and they tend to be more reliable, particularly when you run thousands of samples than sequencing itself. And this gives us a number for each methylation site. We get a number that varies between 0 and 1, where at 0 is, and it can take any value between 0 and 1, where 0 means no methylation whatsoever at that specific nucleotide, the specific C, followed by a G. And again, we measure 850,000 of them, and we get, therefore, 850,000 numbers ranging between 0 and 1. And 1 means that methylation is complete, where the amount of methylation is the measure of how many cells carry or not carry methylation at that specific nucleotide among the population of cells that we used to isolate DNA. So it's really a measure of the proportion of cells that are methylated at a specific methylation site. So using this microarray, we have done a lot of work over the past 10 years. One of the papers that is very interesting to review, it's a meta-analysis of 16,000 people, adults, and 16,000 blood DNA samples, a little shy of 16,000 actually, from 16 cohorts across the United States and North America. This was a study led by Dr. Johannes at Framingham, and the last author was Stephanie London in North Carolina at NIHS, and we contributed to this paper with some of our cohorts. It's a very interesting paper, first of all, because the aim of the paper was to investigate the association between smoking, tobacco smoking, and DNA methylation. And when you look at the differences between ever smokers and never smokers, the results are mind-blowing. We found as many as 7,000 genes, really it's about 1/3 of the human genome, with differences in DNA methylation between smokers and non-smokers. And this really reflects how strong of an exposure smoking is. It impacts our bodies so strongly that our body scrambles to change as much methylation as possible to cope with smoking, to the point of changing the control of methylation in as many as 7,000 genes. What was really interesting, though, is that we had a good proportion of these 16,000 people who had quit smoking. And so that gave us an opportunity to see of these 7,000 genes that are different between ever and never smokers, which one would go back to non-smoking level over time and how quickly. And we found that the changes are very quick. Lots of these 7,000 genes seems to be written with a pencil mark that is very soft, that can go away very quickly. And in a matter of months, more than half of these genes didn't seem to be different anymore from non-smokers in people who quit. So when you compare former smokers and never smokers, many of them didn't seem any longer associated with having smoked in the past. Some others went away in a few years. But what was really interesting is that there was really and these were the other ones were the one in hard pencil. But then we found a few that seemed to be written in permanent ink. And these were just a handful, about 20 of them. They're shown here. These are the differences in methylation in current smokers compared to non-smokers or never smokers, actually. And you can see that you have some genes that have positive association and some genes that have negative association with smoking, current smoker. But you see, these are the years after quitting. And you can see that these genes, they seem somehow to go back a little bit, at least some of them, to non-smoking levels, but many of them remain statistically significant, highly significant, even after 30 years of having quit. So really, I showed you with this paper the dynamic of genes that seem to be written in response to exposures in pencil, soft or hard, and some that seem to be written in permanent ink that never seem to change. Now, there are a lot of fascinating questions about this, how this happens, why does it happen, and so on. And these are really incredibly important questions. But for today's presentation, I want to discuss the opportunity of using these differences to estimate environmental exposures. And this is something that is being done. For instance, there are several groups that have used machine learning, elastic net, or other types of machine learning to predict smoking. The idea is that you get all these 850,000 methylation sites, particularly the 7,000 genes, which is many more methylation sites than that, that are different in smokers versus non-smokers. And you can create an algorithm to create a composite biomarker. For those of you who work in genetics, this is very similar to an epigenetic polygenic score. In genetics, we have polygenic scores. Here we use DNA methylation to combine methylation sites and genes to predict an exposure. The difference with the polygenic scores that are typically only helpful to predict disease is that here we can also assess and predict the exposure because DNA methylation also changes in response to the exposure. And there are several of these. Probably this is only a partial list. It's certainly a partial list. For instance, epi-smoker is the simplest of them all. It just classifies people on ever smokers, never smokers, or former smokers. But there are other over here that also measure the number of cigarettes smoked in average, the cumulative number of cigarettes per years smoked over a lifetime, and also potentially for those who quit years since quitting. So now you have these changes in DNA methylation in our cells that can be used to reconstruct the past exposure and really help with the challenge of time or reconstructing past exposure. And I'll show you more about this and also other types of uses for this type of approach. But I'll show you another interesting finding. So we used one of these biomarkers, which is epi-smoker, which is the simplest, I said. So we classified people, this data in the normative aging study, which is a study in Boston, in the Boston area, predicting, measuring DNA methylation. It's about 700 people followed over time, you see, for at least 14 years after DNA methylation was collected. And we measured lung function loss and death divided by whether people were classified by the epi-smoker as never smoker, light blue, or ever smoker, which is darker blue. And you see here on the left, the incident airflow limitation. You can see how there is a huge separation in the loss of lung function over time. And you can see all-cause mortality is also different based on the epi-smoker. So this is clearly significant, and we expect this. We don't expect things to be different. But what was really interesting, that when we compare the hazard ratios for airflow limitation and mortality based on the epigenetic classifier, epi-smoker, or based on self-reported smoking, we saw that the epigenetic classifier has stronger risk estimates with both outcomes compared to self-reported smoking. And here there is, for instance, not much increase with self-reported smoking, which is a small sample size and people are pretty old, but also raises some doubts about the quality of self-reported smoking. So one difference between these two might be that self-reporting has misclassification, or especially with something like smoking that might present a stigma. People might not be at ease reporting it to the full extent, or at least reporting it correctly. But also potentially the epigenetic classifier is a biomarker that is not just a biomarker of the exposure, but it's a biomarker of the response of the body to the exposure. So it's downstream than smoking itself. You can think about how many things can change the way smoking influences our lungs. You need to think about the topography of smoking, how much of the cigarette gets actually inhaled, as opposed to let it burn on an ashtray or to the air, and also how much of the smoke penetrates deep in the lung, how much it gets into the white blood cells that we are doing. Here we are measuring all of this in peripheral DNA, peripheral blood DNA, and also how the cells themselves respond. So potentially this is really a biomarker that combines exposure and risk. We found the same also with a different type of biomarker, which in consideration of time, I'm going to go pretty fast on, which is a biomarker of lead. As we know, lead is a toxic element that is present in nature that we have used inappropriately in many industrial and domestic uses over the past 78 years, including adding it to the paint, to paint our houses to improve durability. And we were able to create a blood DNA methylation biomarkers that reflects long-term exposure to lead. And you can see the paper here published a few years ago. And what I want to say about this, that in the same way as the smoking biomarker reflects risk better than self-reported exposure, this blood DNA methylation biomarker of lead reflects is more strongly associated to future risk of lead-associated disease than lead itself, than lead measured, for instance, in blood, as we typically do. So I would like to stop here with this, but I would like to give you my vision about the future. So we have all of these incredible biomarkers that can potentially be developed further to measure our exposure. And potentially when we go in 10 or 20 or 30 years to our healthcare provider, they will still probably tell us they want to measure our cholesterol, our blood glucose, our triglycerides, but they will probably also have a panel of the environment like this, a panel that measures using DNA methylation in a single shot by running a microarray on blood, for instance, like the one we use. It will give us a test of the environment. It will tell us about whether we are exposed to air pollution, to tobacco, to metals, to radiation, to alcohol, to a sedentary lifestyle, a low physical activity, diet, etc. And all of this could be combined. So you could get this recommendation from your future healthcare provider saying you are in great shape, you have a healthy diet and great on your active lifestyle. Just to make sure we don't leave stones unturned, we see that your genes have been reacting to alcohol and some harmful pollutants. Can we please discuss? And so this would be a way to have precision identification of people at risk and help tailor messaging to individual people and to individual patients. And down the line, of course, in addition to the exposures, you can also get a sense from DNA methylation of the risk of future disease. And you could try to understand when and how to do any further screening or diagnostic tests based on their risk of disease. And this might seem like science fiction, but in fact it's happening. For instance, this review by Yousefi and others published in 2022 cataloged all the DNA methylation biomarkers of exposure available to that point, where they also put age on the side of the exposure. And you know how there are many biomarkers of DNA methylation age available now. In pink, you see those that can be measured in blood, and this in other tissues, typically saliva or nasal cells. These are the ones that being used the most. You can see there are a few for smoking, more than 20, some for alcohol, BMI, adverse childhood experiences, child maltreatment, bone density, insulin sensitivity, environmental health, and so on. So the message is this is happening. We have biomarkers that are certainly sensitive to environmental exposures, and potentially they are a fingerprint of that exposure. And potentially they can help us to go back in time. The limitations are that they are different from exposure. They are downstream. The specificity to each exposure is still to be determined. And we know that they are sensitive to an exposure. They correlate well to the exposure, but we don't know yet how specific they are, where they might light up also in response to other exposures or other conditions. And of course, they're not tissue specific. If we are using blood, this is the impact of these exposures on blood, probably not on other tissues. And this is important because, of course, if we want to study the impact, for instance, of toxic metals on the heart, of unhealthy nutrition, on atherosclerosis, perhaps blood is just going to give us a partial picture of the impact of the exposure there. And this leads me to the second section and challenge number 2, which is the challenge of tissues. I mean, how do we assess the impact of an exposure of a risk factor across the human body, and whether that exposure or risk factor is important for brain, for the heart, for the liver, for the lung, only, or for all of them at the same time, or other tissues. So this part of my presentation is based on my interest in study the impact of air pollution on cognitive decline and dementia. And that is particularly important because we do know that air pollution, unfortunately, people who live in areas with lower air quality have accelerated cognitive decline, higher rates of dementia when they become older. And we published many papers, including one more recently that shows that even short-term increases in air pollution can temporarily impact the brain health. And of course, these short-term exposures accumulate over time, and the average level of exposure over time is strongly associated with cognitive decline and dementia. And this is an important question, particularly because if you look at dementia and Alzheimer's, they don't start when we are old. They start when we are much, much more young. And this is a diagram from Bucciarelli et al., a book in 2018, where they show, they summarize the evidence that in people who lose the function over time of their mental acuity, those who get Alzheimer's, typically the diagnosis happens when people are 70 or older. Some loss of mental acuity, this on the y-axis is supposed to mean mental acuity. Some loss of mental acuity happens earlier, but it's very hard to diagnose. I mean, it's small, tiny, it's very hard to predict whether this is really different from this. But what is interesting is that if you look at the brains of people who die for other causes, like accidentally, earlier in life, in their 50s, their 40s, and even their 30s or 20s, you can often see the pathology of Alzheimer's already in their brain. And this suggests that the etiology, the pathogenesis of Alzheimer's starts very early in life. And this creates a challenge because we have something that manifests itself here, but we would love to measure the impact of the environment here, anywhere here, earlier than when the memory issues start to emerge. And particularly, we would like to see the impact directly on the brain. So a solution to that may be extracellular vesicles. Extracellular vesicles are membrane-bound, small, nano-sized vesicles, usually smaller than 300 nanometers. They are found in any human biofluids, plasma, serum, saliva, urine, CNS fluid, and so on, breast milk, ovarian follicles, and so on. And they are believed to be now a previously unknown signaling system between cells. Cells release, this is supposed to be a cell, cells release the vesicles here, and they go from here to other cells. And even in the circulation, they go to other cells remotely. In a way, they operate, we believe, not unlike an endocrine mediator, like a hormone that is released by a gland and released in the circulation and homes to any other tissue around the body. It can change gene expression or functioning in the recipient cells. The same way, these vesicles can be taken up by recipient cells, and the materials that are inside, the cargo, can get released into the recipient cells and change the biology of the recipient cells. Particularly, we have been interested in microRNAs, which are short, non-coding RNAs. They do not code for a protein, but they can regulate gene expression. So they can regulate specific genes and regulate whether they produce a protein or not. And so we have a situation here where the microRNA is included in a vesicle, released outside the cell, can go in the circulation, and potentially vesicles that come from the brain, or they might go to the lung, might go to the cardiovascular system, or vice versa. Vesicles released from the lung in response to air pollution can travel everywhere and influence responses and diseases elsewhere. So let me give you another analogy here about how we think about extracellular vesicles. And this one of my students, Rhodos Rhodostenis, came up with this idea that in a way we need to think like our cells are emailing each other. And you can think there is an email from the lung epithelial cells, which are their first cells likely to encounter air pollution, and they might write to blood neutrophils and copy lung alveolar cells and brain microglia, and they would send an email with the subject of response to air pollution and might say, "Dear neutrophils, how are you doing? As you are fully aware, we have been exposed to higher levels of air pollution than usual. I need your help. Please see what I need from you. I'm going to send you an attachment with many microRNAs that are going to tell you how to change your gene levels now in this special situation." In a way, this is a little funny, but we do think that the cargo, the attachment, really in the vesicles can do that. So I'll show you some results on, particularly on how we can get, how sensitive these biomarkers can be and how associated with cognitive decline and dementia again, in the normative aging study. This study included the sub-study on vesicles, including 656 participants who were active in the study after 1995, which is the year when they started to bank plasma samples that we could use for this. They come every 3-5 years for follow-ups, and in these plasma samples, we found 381 microRNAs in vesicles that were found in at least 70% of plasma samples. And we also had a measure of cognition, which is the mini mental state examination, which is a questionnaire-based measure of mental acuity. And what we found, and we had this measured every 3 to 5 years, so we used the baseline microRNAs in EVs to see how that would predict changes, decline in mental acuity over time. And we found very many microRNAs. We found many that were negative, many that were positive, and strongly associated with both higher mental acuity or lower mental acuity. And this really shows that the test has potential, but again, what I didn't say is that this was not studying the brain specifically. This was vesicles coming from plasma, regardless of where they came from, total vesicles. So there is a minority of vesicles that we expect to come from neurons and from the brain, but we were not able here to differentiate those ones. So what we are now doing, and this is funded by an R35 to my lab, we can isolate the vesicles in plasma that are released by neurons. And particularly we do that because vesicles carry on their surface proteins that come from neurons. This is not just neurons from the brain, it's neurons everywhere, though these vesicles pass the blood-brain barrier. And of course, I mean, we can use antibodies to pull down the proteins, and then we can measure the RNA only in those, or almost exclusively in those vesicles that come from neurons. And this is a huge advance for us. We can do that not just in neurons, but also in other cell types. For instance, we are now based on this R35, we are now measuring, we have methods to isolate neuron EVs, but also astrocyte EVs and microglia EVs in plasma. So they are coming from there. We are working on oligodendrocytes. We have methods already for lung epithelial. We have methods for liver. We are working on adipocyte. And down the line, we want to do all of this. And another interesting development is that we can also use analytic chemistry to measure chemicals, which is very interesting for my research. Potentially, you can also measure nutrients. You could do metabolomics, for instance, on vesicles, and measure really the signals in the tissue that is most interesting and appropriate to your question, rather than overall in blood. This work that has been led by HNRCA, who's an assistant professor here at Columbia. Of course, there are a lot of challenges. There are, and I'm going to wrap up here, there are many exposures to investigate. If we want to, and we started only with a few of one, if we want to create lifetime molecular biomarkers, like in the first part of my presentation, we need to calibrate those on lifetime data. And for many exposures, we don't have that data. And finally, these efforts need to undergo validation across different populations to show that they're valid biomarkers universally. So this is all I had today, and I want to thank my lab team and collaborators. Many you saw them on the slides. And of course, the NIH has been very generous to us, and this is the funding we currently have in my lab. And thank you so much, and I'm happy to take any questions.
Well, thank you very much for a really interesting presentation. And we do have some questions. And the first question is from Richard Alpert, who asks, are there any unifying factors, characteristics of the genes that are altered by smoking that either did or did not return to baseline after quitting?
Not that I know. There are, even looking at the gene architecture and function, it's very different to identify them based on their a priori information. A couple of those, for instance, one that you might have noticed is the HRR, which is the aryl hydrocarbon receptor repressor. That is the gene that is also, that shows the biggest change also. So it's not only the one that is most persistent, but it's also the one that has the largest and the most significant change. So in part, they seem to be hypersensitive, the ones that remain forever, but really it's difficult to classify them based on function.
Okay. Next question comes from Larissa Constantinow. Is there any evidence of an effect of e-cigarettes on the epigenome?
Oh, there are a few studies that come up. They're small in size, and clearly we expect DNA methylation in the epigenome to be modified by e-cigarettes. We do have, including in my lab, studies on some of the components of e-cigarettes. For instance, we have a lot of studies on metals, and unfortunately, e-cigarettes tend to release toxic metals in the lungs of vapors because the coil gets dissolved. The metal coil gets dissolved in the smoke, in the vapor. And so really, you get a lot of toxic metals like cadmium that we know that is a lung carcinogen that gets into the lung. So there are some papers on this, but it's still a field that needs to be explored. If you're interested in vaping, you should go for it. I'm happy to discuss collaborations.
The next question comes from Eileen Kennedy, who asks, how solid is the evidence on the 451 genes on unhealthy diet that can be activated?
So sorry, I made my example was really fantasy, so I made them up completely. So there are many, and probably there are more than 451, and there is a lot of evidence on nutritional. There are a lot of nutritional epidemiology studies, including meta-analysis. I'm happy to send you papers your way. If you send me an email, I'm happy to send you papers of the meta-analysis I know on nutritional factors, diet, and DNA methylation. But the numbers I put on those slides were completely made up. They were about in the ballpark of the numbers I would expect based on the literature I know, but they were not based on actual literature.
Okay. Next question is from Jean Dangwang, who asks, are there any longitudinal cohorts to study if cigarette exposure at earlier age, example, younger adults, could affect epigenetic classifier or those genes that never return to non-smoker levels and predict aging, example, decline lung function in older adults?
So there are a few studies of birth cohorts, for instance, ALSPAC, where they have measured DNA methylation in children at birth in their cord blood and also in blood in their childhood. And they do have data now, I think, for 30 or 40 years. So it gets to that level. One study that has a lot of potential is CARDIA, which is a cohort of adults in the United States. It's 3,000 people with DNA methylation data already completed, and I think it's available in TopMed for those of you who are interested. So you can download it for free. This methylation data has been done every 5 years for 20 years, and the participants were 18 to 30-year-old when they were recruited, and now they are approaching the 40-year mark of their follow-up, so they are 58 to 70. So that's a study where some of these questions can be tackled, but I don't think there is yet a study published addressing the question you are asking.
Okay, and the next question comes in from Roger Fielding, who asks, how do you deal with the synergistic effect of multiple types of exposures on the methylome?
Yeah, that's a great question. At the moment, we haven't, and there was really the situation, the limitation that I described about specificity. Typically, in these epigenome-wide association studies of DNA methylation, we run a multivariable regression where we adjust for several factors to exclude the influence of other factors. But I think your question also goes to the idea that there might be interaction between exposure, not just confounding. There might be effect modification. And that is a great question. We are now starting to use different methods based on data science that can lead us there. There are different, in our field, we call them mixture analysis, the idea that you mix together different exposures. Like when you do animal experiments with chemicals, you can give them a mix of chemicals rather than one chemical at a time. And this has now come into some data analysis methods in epidemiology where you consider multiple exposures at the same time. But the field is just starting. And I think for those of you who are biostatisticians, it's a very active area of development. As the exposome becomes more of a mainstream topic, there is a lot to be done about assessing the impact of the exposures, of multiple exposures at the same time.
And the next question comes from Chao Lai, who asks, given environmental factors affect multiple DNA methylations that are overlapped, how can we identify unique DNA methylation markers for a given environmental factor?
Yeah, exactly. I mean, we hope they're unique because they're usually made of tens or even hundreds of methylation sites. So we hope that the combination is unique to that exposure, but we know how there are exposures that activate the same pathway. So I think the jury is still out about how specific these methylation biomarkers are. Though they seem to be specific enough based on what I've seen to time, but really there are to date, but there isn't really been a lot of studies seeing whether a smoking biomarker gets also activated by air pollution, for instance, or by alcohol for that matter. So this is really something that needs to be done.
Okay, and I'm seeing no more questions in the queue, and the fact that we've reached the hour of 1 o'clock. I'd like to thank you again for a really fascinating presentation.
Thank you so much. I'm very happy to be there.
Thank you. That was terrific. Love your slides.
Thank you. I learned how to use PowerPoint recently.